Go ahead.
So, hello, I'm Eric Rottinger from Mark Martindel's lab at the Kiwala Marine Laboratory in Honolulu, Hawaii. So today I'm going to present you a database that we developed together with Matthias Omerstadt, a former postdoc in the laboratory. And the initial point was that we had a lot of data that were published and unpublished within the laboratory about various marine invertebrates, and we tried to find a way of storing those data within the lab. And this grew and became bigger, and we thought that it might be a useful tool for the whole VID00054 community. So what we did actually is we created a web-based database where people can upload published or unpublished gene expression data from various marine invertebrates. So here we have an overview of the few species we have already in there. So there are bryozoans, ACLs, sea urchins, ctenophores, Nematostella, and brachiopods. And this database you can actually query either by gene name, by expression domain, or by developmental stage. So how that works actually, here we have an example of the Nematostella OTXB gene. You can ask to get information for this gene. It shows you the whole expression pattern in an image view, and all those small errors actually show you that there are different views. And it also shows you the expression data in a grid view, where the black data means there's no expression, the white data means that there's no information available, and the green dots tell you that at the early gastrula stage this gene is expressed in the animal pole in Nematostella. The database also suggests you, based on those information that have been added by the user, genes which have a pretty similar expression pattern. So in this case, OTXC and OTXA have a strong similarity to that expression pattern. So in the next figure you see actually when we select those two genes and compare that to the initial query, you get a grid view where the green dots, the green square, show you that all three genes are expressed at that stage in exactly the same domain. So when you click on that, you actually see the real expression patterns on all three genes, and here you clearly see that that gene, or those three genes, are really expressed in the same domain at the same blastocyst stage. Then if you want more details, you can click on the individual image, and it gives you more information about who did that, which laboratory did that, and how that gene is expressed. To make that tool a little bit more useful for a broader functional analysis, you can also look at individual stages, how a gene is expressed. So in this case, we look at the blastocyst stage for all the genes that are expressed in the animal hemisphere, which is the presumptive endoderm in cnidarians. So this query gives you a list of genes that when you compare, actually, you can visualize at one time that all those genes are expressed in the blastocyst stage. So in this case, we have OTX, Sproudi, SMAD15, and FOXP that are all expressed in the same territory, which suggests that this is a syn-expression group of genes that are probably involved in the same biological process during development in the animal, and that can suggest some genetic interaction for further functional analysis. What you can also do is actually not only looking within a single species, but you can also look how genes are expressed across species. So here we looked actually in the database for OTX expression of all the genes that have been added to the database, and we get a list of about 10 different genes that are expressed in cnidarian, in hemichordates, in parahylae, in corals, and in Terebrutalia. And then, similar to what I just said above, you can click and select individual squares, and then you can see the expression patterns where the genes are co-expressed, or by clicking at the individual names, you can see the expression pattern of each individual gene that has been added. So the final feature, what we wanted to do is that it's not only for wild-type expression patterns, but that it's also for functional analysis. So in addition to the wild-type experiment, you can add drug treatment or lithium treatment in that case. So this is a sea urchin expression pattern, And the experiment that we added is a lithium treatment, which is a way of activating the Wnt-beta-catenin pathway, and we see that this gene here is overexpressed in lithium treated compared to control embryos. So this is also a way of storing data of functional analysis. So what we wanted to do is that, or what we hope that this database is going to do, the people can submit data that are already published, or they can submit data that are unpublished, but that are accessible to people from their own laboratory, or they can invite groups from external labs to share those data to create some collaborative groups. So finally, this is the status of the database right now. We try to implement to that the embryological developmental database that explains more in detail the individual developmental stages and expression domains from those species, and we hope that this is a tool that will help to understand those expression patterns or the development biology of different animals.
Very good. And people can see this at
kahikai.org.
kahikai.org.
And then on the first page there's a small button that links you directly to that database.
Very good.
Thank you.
Thank you.